Good afternoon, everyone. I am Dimpi Singh, faculty in Mahatma Gandhi Institute of Healthinformatics. Today I am going to discuss about nucleotide databases. What are the nucleotide databases? As the name indicates that nucleotide databases are the databases which contain the information about the nucleotides. As we know that nucleotides are the sequences are the language of DNA and RNA, and we can say that the nucleotides are the monomer of DNA and RNA. So there are three major sites for finding information about nucleic acids on the web, and all of them contain basically the same information. The methods and databases that you will want to use will depend mainly on how much data you want and in what form. It means the sequences which you want to access from the webs is depend only on your research work. So the databases, mostly there are different type of nucleotide databases, but the most important databases are three, that is EMBL, European Molecular Biological Laboratory, GenBank, and DDBJ, that is DNA Data Bank of Japan, are the three primary nucleotide sequence databases. They include sequences submitted directly by scientists and genome sequencing group and sequences taken from literature and patents. As primary databases contain the data which are directly received from the experimental labs. For example, GenBank has currently 17 divisions. There are no legal restrictions to use the GenBank to access data from the GenBank. However, there are patented sequences in the databases. So from the GenBank, we can get the sequences of any particular gene, any particular nucleotides which we want to work on it. So there are different type of biological databases, primary databases, secondary databases, and derived databases. So here we are discuss about nucleotide databases. So you can see that the nucleotide sequence databases contain three databases, mainly GenBank, DDBJ, EMBL. Protein sequence databases, SwissProt, PIR, GenPept. But we are discussing about only nucleotide sequence databases in these presentations. So GenBank databases. Basically, GenBank is available on the NCBI. GenBank is a tool which is enable to find out the sequences, to access the sequences from the databases. So GenBank is the most complete collection of annotated nucleic acid sequence data for almost every organism. We can filter the data from the NCBI. If we want the Homo sapiens sequences, then we can do it. And if we want that musculus sequences access, then we can do it. The content include genomic DNA, mRNA, cDNA, ESTs, high throughput raw sequence data, and sequence polymorphism. There is also a GenPept databases which is mostly used to access the peptide sequences. It means amino acid sequences. Although a small number of the amino acid sequences are derived using peptide sequence techniques. So submitted sequencing to GenBank is also very easy and is required by most journals before articles pertaining to the sequence are published. You can submit your sequences which are output of the experimental techniques by www with Bankit. How to search GenBank? GenBank is a tool which is integrated with NCBI, and NCBI is the National Center of Biotechnological Informations. So there are different type of databases like nucleotide databases, protein databases, gene sequences, SNPs, and PubMeds. So there are two ways to search for sequence in GenBank. One is using text-based keyword similar to PubMed search. If we want to search out a particular gene sequences, then what we can put the name of gene as a text in a search bar and then click on search. Then we will get our result. So one is using text-based keywords similar to a PubMed search, and the other is using molecular sequences to search by sequence similarities using BLAST. So if we want to align two sequences, the alliance sequence which we get from our experimental techniques, then we can directly put the sequence into the alignment tool, that is BLAST, which is responsible for the checkout similarities and dissimilarities between the sequences and to find out the phylogenetic relationship between the sequences. So GenBank sequence formats. So when we will get our output by the GenBank databases, so then our output categorized in three categories. First one is header, second one is features, and third one is sequence. So first header define the description of about the gene, about the organism, about the species, about the taxonomy. It means header contain all the information about the gene, like organism, like sequences, species, like authors names, PubMed links. And the second category is second one is features. So features included the data about the gene which contain genes, exons, introns, CDS, conserved domain sequences, SNPs, and the proteins encoding by the particular gene. And third one is sequences. And third one sequences, the nucleotide sequences are there in a faster format. So to search GenBank effectively using the text-based method require an understanding of the GenBank sequence format. GenBank is a relational database. However, the search output for sequence file is produced as flat files for easy readings. So when we get our GenBank result, it is very easy to reading, it is very easy to analyze because it is a text based, it is a English like language, so we can easily read the result. How many exons are there, how many introns are there, how many conserved domain sequences are there in a particular sequences. So the resulting flat files contain three sections as I earlier defined, that header, features and sequence entries. There are many fields in the header and feature sections. Each field has an unique identifier for easy indexing by computer software. Understanding the structure of the GenBank file help in designing effective search strategy. So it is the example of output of GenBank databases. When we put any gene name on the search bar and select the nucleotide databases, then we will get this type of output. And so the header part here, header define the locus of the gene, define the definitions, it means the title of the gene. Accession number is a unique number. Unique number, it means the unique number of gene. Version ID and sources, organism and publications, authors, titles, and there are PubMed links included in header. In second, we can see that features included sources, gene, and the gene which is responsible for particular protein, gene encode protein is also included in features. And third one is sequences. So we can see that sequences here. For a nucleotide sequence that has been translated into a protein sequence, a new accession number is given in the form of string of alphanumeric characters. So accession number is the number which is uniquely identified of a particular of a specific gene. In addition to the accession number, there is also version number and gene index number. The purpose of these numbers is to identify the current version of the sequences. If the sequence annotation is revised at a later date, the accession number remain the same, but the version number is incremented as the GI number. A translated protein sequence also has a different GI number from the DNA sequence it is derived from. So alternative sequence format. if we will talk about GenBank, so there are two type of formats to give us information. First one is GenBank format and second one is FASTA format. In GenBank format, as we can see that the all of the information about a particular gene is given. In FASTA format, only sequence are there. So in bioinformatics, FASTA format is a text-based format for representing either nucleotide sequence or peptide sequence in which nucleotide and amino acids are represented using single letter codes. FASTA is one of the simplest and the most popular sequence format because it contains plain sequence information that is readable by many bioinformatics analysis programs. It has a single definition line that begins with a right angle bracket followed by a sequence name. Sometimes extra information such as GI numbers and comments can be given, which are separated from the sequence name by a symbol. So this is the FASTA format sequence. If we click on FASTA, then we will get this type of output, only sequence with the definition and title. So the extra information is considered optional and is ignored by sequence analysis programs. So the plain sequence is standard one letter symbol start in the second line. Each line of sequence data is limited to 60 to 80 characters in width. Thank you.